Alright, so hopefully you can see my screen. So this is I've been looking at the L1000 data and looking particularly at this gene pairing. Just a quick recap. A single experiment measures 1,000 genes, but it only uses 500 different colors of beads to do that. And so each bead does double duty. In general, there's approximately 30% of beads are marked to measure the expression of one gene, and 70% are marked to measure the expression of a second gene. And I believe the idea was to pick two genes that are separated enough in expression levels that they would be easy to deconvolve once these beads get measured. On the right on the slide is just kind of an example of a toy example of what you might see in terms of the distribution of expression levels measured based on the beads that you're sampling in an experiment for a particular pair of genes. But unfortunately, it seems like sometimes it's not clear where these peaks are. They get put together, or there's due to sampling, one peak doesn't show up very strongly. In addition, there's a gene flipping issue where the expression level for gene 1 can be assigned to gene 2 and vice versa. So I wanted to look and see, how widespread is this issue? What do the gene pairs look like? And this is all kind of exploratory, so I'd be glad for any questions and comments. So the first thing is, I'm looking in particular for this presentation at a single cell line, A375. I'm looking at all the untreated experiments in bead batch B4. And I do that just to narrow it down to try to control some of the variability. This results in about 630 experiments. So what you'd hope to see is something kind of like this. So this is gene PSMD9 on the x-axis and PEX11A the y-axis. And you can see there's a nice cloud here. There's a few points here which I'm not sure about. And then there's this observation here. And this one, it looks like if the gene values were switched, it would fall right into this cloud pretty nicely. So I would think that maybe that was just one where the gene pairs got flipped. So this is, like I said, one of the nicer examples. This is one of the more pathological cases that I found, where you get multiple clusters of data. So it's not just, we're measuring these two genes with some random noise. There's this cluster that appears here, and then this one over here. And these two clusters actually look very mirrored. And I'm not sure if that's this gene flipping issue or something else. You also get a fair number right along this 45-degree line, which is x equals y. So that's when the two genes are unable to be deconvolved for whatever reason. The interesting thing about this particular pair of genes, and I'll show you a little bit later, is if you look at other bead batches, they all fall into this lower cluster. And these other clusters don't exist in those bead batches. So my question is, what's causing this? Is there some way to control for it, or even possibly to adjust the data so that it makes subsequent analysis better? Here's another example of a different sort of question that I have. So it looks like there's two clusters that are decently mirrored, but is that the case, or is this actually one cluster that just happens to fall around this 45-degree line, the x equals y line? It's interesting because there's only a couple points that aren't able to be deconvolved, and then there's this gap, which I think is common in the data. If the peaks are too close together, that it just doesn't happen in the data the way that it's set up. So you get this gap. So my question here is, is this one cluster that kind of goes across the line, or is it actually two clusters, and are some of these flipped? Like I said, I don't know enough about the biology side of things to know, what to expect. I mean, from my understanding, I would expect all the points to be predominantly on one side of this line, where one gene is expressed more highly than the other. So insight into this type of artifact, I think, could be really helpful. And then just to show an example of a pair that for bead batch B4 just didn't work very well. You get a bunch of points right along this line, the 45-degree line, and kind of the whole range of expression values from close to 0 all the way to 15. So what I would assume is something about bead batch B4 that just didn't work for this pair of genes. There's a little cloud on the right, and maybe a couple points that have flipped data. But predominantly, it just looks like this bead batch failed for this pair. So again, I'm not sure why this should happen, or what necessarily is the best thing to do about it. So here's the examples I've shown along with a couple others, and you can see various levels of artifacts in the data, for lack of a better term. Some that maybe there's some mirroring, maybe there's multiple clusters. The lower right plot here looks like it exceeds the maximum expression level threshold for most of the experiments. So if you look at this for more than just bead batch B4, I did, I think, the three largest bead batches, which are B3, B4, and B7, plotted here. B4 is in green, B3 is red, and B7 is blue. And so here's the multi-cluster example, and you can see almost all the blue falls into this lower cluster, and most of the red, although there's a little bit. But in general, it looks like, at least for these examples, bead batch B7 seems to be a little bit more consistent across the data. One thing to notice here is this left center one is the x-axis is one that I've talked about in the previous discussion meeting. It's ERBB3. It has two peaks if you look at just the expression level of that single gene. And bead batch B7 is the one that kind of shows up as that secondary peak. And here you can see that, yes, it is shifted. But in most of the other cases, it looks like B7 is more consistent. So, in that case, is that true here? So those are just different things that I'm trying to get a handle on and understand what to do with. It's just to show that it's not just that cell line. Here's another cell line that has with the same. It's the same set of pairs, gene pairs, so beads, as the other plots. But again, you still see this multiple clustering effect, and some cases where you get a lot of points right on that x equals y line. So again, my main questions, and I put this up on the discussion group, the Google group, as well. It's just so we can, start having some of this documented and get people's responses as they think about it or whatever. But my main questions are, what's causing these artifacts? And I've done some looking, and for example, for this one, there are single plates, so there's multiple untreated controls on a single plate, and there are single plates that have some in the lower cluster as well as some in the higher cluster. So it's not just an artifact of different plates were done differently. It's actually experiments on the same plate show up in the different clusters. So I've been looking kind of all over the place to try to figure out what's going on. Is there evidence of this gene flipping? I believe there's some correction done for that already, but is this more evidence that they're still flipping this slipping through, and how much? Is it just, do we expect just a few points, or are there cases where maybe 10, 20, 30% of the points get switched? In cases where you have kind of the mirrored clusters right around the x equals y line, why are there so few points on that line if it's supposedly a single cluster? And then what do we do with that? And do we ignore it? Do we decide some experiments are just bad and throw those out? What's the best way to handle that in order to make the subsequent analyses better? So one thing I looked at, and I know that this has been done, is subtracting off the controls on a plate from the other experiments, the perturbation experiments, and dealing with kind of the difference values. I know Shannon did a lot of that with his cosine distance stuff, and it seems to be kind of at least somewhat the common method for dealing with some of this. And so, is there a question?
Hello, this is Adrian Raftery here. I've just joined.
Yep. All right. So I see clusters in the untreated data. What happens if I look after I adjust for these untreated experiments on the perturbation data? And so one data set I looked at is knockdown experiments for gene ERBB3 in this cell line. And so here I take the gene expression level for an experiment. I subtract out the average of all the untreated experiments on the same plate, hopefully getting rid of some of the kind of plate effect that we've seen before. And so here is one example. Excuse me. Neither of these are the knocked down gene, so we don't necessarily expect them to change overall. And you can see the upper left plot is the untreated data from the plates, and this is actually colored by batch. And there does seem to be at least some clustering by batch for this pair of genes. And then the lower left is the data from the knockdown experiments for this pair of genes. And you see it corresponds fairly well with the same clusters. The lower right is once it's adjusted for the plate untreated data. The 0, 0 point is right here. You see that it does, there's a cloud kind of centered around there. Looks like perhaps this blue batch has more variation than some of the others, but that's kind of what we'd expect. There's no clustering going on here. This is the example of the pair that did have the clustering shown before, and for example, this green has a large portion here, but this is one of those other clusters that had kind of a mirror up here before, but for this particular data set, they show up here. So here's some clustering before, and if you look at the knockdown data, you still see a few points that show up there, but predominantly they're in the lower cloud. And then when you adjust, again, you get this cloud around 0, and you don't get the clustering that you see in the untreated data. So I think that at least is encouraging, that the clustering, at least for this, isn't strong enough to induce clustering once the data has been adjusted on the experiments. It might be adding extra variance to the data due to the fact that you're taking an average, maybe up here, not within a tight cluster, but overall it doesn't seem to be too bad in terms of inducing clustering or pulling things way out of where we might expect them to be. This is actually the target gene on the x-axis. ERBB3 is the one that is supposedly knocked down in these experiments. And you can definitely see that that's the case. The green kind of smears it to the left compared to the untreated data. And when you do the differences, the 0, 0's here, so you see most of the data is on the negative side. So it does look like this gene is knocked down. The vertical is not as much signal, which we don't necessarily expect. Again, it's not the same gene. But again, this is encouraging just in seeing that, in this case, the knockdown is does appear to be doing something. So, kind of summary, no clear clustering after I do this adjustment, maybe introducing additional variance due to the clusters in the untreated data. I do think there could be potential benefit from identifying some of the issues early and fixing them before doing the adjustment. I'm not sure to what extent that might be helpful. I think there are some benchmarks that might be able to be done. And what I'm currently working on is kind of identifying, is there a way to methodologically adjust some of these data and identify flipped points and bring them back into a more reasonable spot. But really, that's what I have. I'm really open to any suggestions and questions. What has been done? What can I think about? What might be causing these issues? So, yeah, that's what I have. Thank you.
So this is Mark here from University of Montana. Can you hear me?
Yes.
Is it possible that these mirroring or these sort of clusters are actually real, and that there are kind of different states in the cell that reflect different expression patterns of two genes versus one gene versus another? Some are sort of linearly related, like the one in the middle on the right, but there seem to be a number with clear clusters, sometimes 3 of them.
Right.
Could those be states of the cell or something? I mean, do you think it's really artifact, or do you think it's biology?
I really don't know enough about the biology of things to say. I've thought about that. One issue is, if that were true, it's only showing up in this, for this multi one up in the upper right. It's only showing up for one of the 3 largest bead batches. I'd expect, to see something similar in the other bead batches. So
Could I ask, were these different bead batches done on the same cells at the same time, or were they done on different days?
I believe they're done different days. There are different plates and different batches.
The reason that I ask that is because I've seen microscopy where they're looking at phosphorylation events or endocytosis or a number of other things, and there's profound heterogeneity even within the plate itself, so that cells that are in clumps look completely different than those that are isolated. The ones that are isolated have much more active signaling, migration, endocytosis, as you might imagine, because they're moving, and the ones in clumps are sort of dormant. And this could be, if your cells are more or less dense or confluent, then that might affect the results as well.
Yeah, I can totally see that is possible. I don't know how to distinguish between those things. One thing I did look at is if you look at which points fall in which clusters, and you kind of color the other gene pairs by that, there's no obvious correlation between the clustering and other gene pairs. So, if it's in, this cluster here, those particular experiments aren't necessarily going to show up, say, in the center one in this top smaller cluster. They're still distributed everywhere. There might be some kind of common patterns, but not that I've really seen across gene pairs, if that makes sense.
Yeah, it's puzzling.
Mm-hmm.
So I guess, Chad, and this is Mario speaking, I hope you guys can hear me. You remember I did have a theory about these things, and I guess I didn't, was not organized enough to reply to your message in the discussion group, but I will write this down and send it as a follow-up, and then people can comment on both of these, I guess. So maybe I can introduce this theory. What do you think?
Yeah.
Okay. So, and I have a couple of plots that go to some extent to what you were doing with subtracting the experiment effect. I assume that was a plate level average or some sort, yes. And how, the cluster on level 3, this is basically level 4, then data, how these clusters in level 3 map to level 4. So I will make myself a presenter at this point. If you're okay, I can switch you back later.
No, go ahead.
Okay, so this is a way less sophisticated looking plot, but it should be the same data that Chad showed. And so on the first one of the first plots that showed basically these 3 clusters of expression levels and level 3 data. And one thing is that he also, I think you, Chad, mentioned that this cluster down here was reproduced in other batches, right?
That's correct.
But this one was not. So my hypothesis here was that we actually have So this gene IKZF1 is actually expressed on at relatively low levels. And if you can see the expression levels of the gene on the x-axis is fairly uniform, right? In both of these clusters, it doesn't shift all that much.
Yeah.
But this one really, y-axis shifts a lot between these two clusters and this third cluster. So basically, my hypothesis is that this gene overall is low expressed, and often we just don't. It's below the detection limit, at least in this bead batch. So what happens is that whenever its observations are below detection limit, we get this picture here, meaning that all of these measurements are actually measurements for the CLTC gene, right, for the x-axis gene. But they are forcefully, by the algorithm, either split in two different measurements, right? Because that's what the algorithm does. It takes measurements that it has that are observed and tries to split them in two clusters that are approximately 2 to 1 in terms of numbers of measurements in the cluster. And then on the x-axis are the ones that could not be separated, I guess, samples where it cannot find two different clusters. So this picture is fairly typical, right, of pairs of genes that have similar expression level, I would say. Or even if not necessarily similar, but any pairs of genes that were measured on the same bead. Well, no, I guess the ones with similar expression level. So that would be my hypothesis, that basically here these are all expression levels related to CLTC, and these are the ones where we actually measure both of these genes, and then they are well separated and everything is fine. All right, and so the next thing that I'd like to show you is basically how these two clusters of points, 1 and 2, behave in the level 4 data. This would be equivalent to what you called experiment adjusted, I believe, measurements. But it's not just that. I guess the algorithm that is used to generate level 4 data also adjust for some of these flipping effects. So there are a couple of steps that are done there, so it's not just simply subtracting the mean behavior per plate, but there are a few other steps that are done. And so what happens is so this is the cluster that corresponds to this cluster in level 4, right? So these are now these Z statistics in, oh, I'm sorry, that was level 3, this is level 4. And the other one, this is the cluster that corresponds to the other group here. And if you think of the theory, which ones should create outliers for the gene on the y-axis, it would be this cluster, right? Because these clusters have nothing to do really with the observations for this gene. They're just kind of split observations for the gene on x-axis. And if you look at the level of deviations of these Z statistics, there is a clear group starting somewhere here around 3 or 4, and then going all the way to 30 in terms of positive Z statistics. Or so these would be really, really unusual observations. And it would basically correspond to deviation between what you see at this level of expression for this gene versus this level of expression for this gene. If you look at this other picture, the deviations are much smaller, right? The scale goes up to 8, and vast majority of them are really less than 2. So it would kind of maybe indicate that this group behaves nicely when it's turned into Z statistics, and it represents basically native behavior of this gene, versus if you do it, if you look at this group, it produces these large outliers because simply these observations are not related to this gene.
Right.
So that's, I guess, one thought about these things. The other is, you know, again, what do you think of basically using level 4 data versus using level 3 data? And then the last question is, would it be worthwhile looking at level 2 data for this one example and see what's happening there? I mean, I am fairly convinced that this is not biological behavior. But again, I'm a statistician, so maybe there is a reason, biological reason for this.
Yeah. I think personally I like looking at the level 3 data because I do see the artifacts, and just in terms of helping me understand what's going on. Because part of it is when you get to level 4, if you don't look at the level 3, you have your two other plots basically overlaid, right? You can't tell that one came from one cluster and one came from another, right? Most of the outliers, yes, would come from that secondary cluster, but overall, it's not really telling you that you can differentiate between the ones kind of around 0.
No, I agree. I think that this information that you get of possible pathological behavior is important, because I'm not completely convinced that whatever is done with data going from level 3 to level 4 actually accounts for that. So in this case, I think it would be really worthwhile knowing that these observations may not be right.
Mm-hmm. Yeah, I mean, I don't know if this is in the level 2, but I mean, it would be interesting to know, are there, how many of the measurements were, out of range, or no expression level, or, something like that. So, you should be able to tell if that's why it's causing these, other clusters, right?
Right, I think.
Yeah.
Yeah, I haven't looked at level 2 data in probably 6 months now, or maybe even longer. But I would guess that this information is there. So.
Yeah.
Any other thoughts? Anybody?
Hi, could you hear me?
Yes.
Yes.
I'm thinking from practical perspective, if you could propose some ways to somewhat address these artifacts, and then give us, have some new processed signatures and benchmark them and show they are better, maybe we can build a better pipeline to present all this data. Yeah.
Yeah, I agree. Yeah, that's one thing I'm trying to work on. How do you do this kind of across everything, and do it in such a way that it does, doesn't overcorrect things that shouldn't be corrected. But yeah, I definitely agree, and I've been working on different ways to look at this data, try to figure out how can we make it better. I don't have the benchmarking I'm not fully, I don't have a full understanding of that, but I think it would be good to do that and not just kind of blindly fix the data without knowing what it does further down.
This is Kai Young from University of Washington. So I'm wondering in the audience, does anybody know what are the geo expression profiles that Broad has used to generate the interpolation, the regression coefficients? Because if we know what are those geo expression, the profiles, we could probably use that as an external kind of a benchmark to gauge whether one of the genes are supposed to be, in general, lowly expressed using Mario's hypothesis.
Yeah, well, that was, I think that data set was available, at least the data matrix, at some point. So I guess we could again investigate whether this information is available. The other thing that could maybe be valuable is that there is in the metadata for these experiments, there are baseline levels of each gene for each cell lines that were measured, I think, in this CCLE data set, or so it was measured using an alternative technology. I don't recall whether it was RNA-seq or microarray, but there is this information in the metadata that can be downloaded through APIs.
That would be extremely helpful. We would really appreciate a pointer to the CCLE data. Thank you.
Okay, so if we will extract this information, or at least point you to the right field and the right API, and post this on the in the discussion group.
Great.
So I guess that would be. We still, I think it's worthwhile thinking of what we're going to do for benchmarking, or what additional things we can do for benchmarking, and that's another topic, I guess. But the benchmarking can come in different shape and forms. One is benchmarking of the whole signatures, right? Or do we connect the signatures that need to be connected? The other one is benchmarking at the gene level, and which genes are reliably measured and which were not reliably measured.
How do you benchmark on gene level? How do you decide which one is reliably measured?
Well, I think that's what Chad is doing here to some extent. And I don't have a very good idea, Chanan, at this point, but maybe going through these baseline measurements that we know, and then look at clusters that show up like this in level 3 data, and how they finally translate into this level 4 z-scores, would give us some idea of which expressions are reliable and which expression levels are reliable and which are not. So Chad, do you have any idea of how to systematically kind of do some of these examinations that you are?
One thing I've looked at is doing a model-based clustering on the gene pairs, and then kind of even looking to see, how many clusters are there, how many of the data points show up in kind of the dominant cluster or even the dominant group of clusters that may overlap really well. Because then you get some kind of, 90% were in this common cluster, or 95%, and that's signifies that at least the data is consistent. So I've been looking into that. I think I found the expression levels, like the baseline expression levels. I'll definitely be looking into that to see, what I might expect for some of these pairs in terms of which one is more highly expressed overall. I think that could be really helpful as well.
Any other thoughts, questions?
I'm doing the benchmarking, so I can share the result after I get it done.
Shannon, I had really trouble hearing you. I thought I heard you said you were doing some benchmarking and you could show it to us when you're done. Is that what you said?
Yes. Mainly from level 4.
Okay. But that would not be now, right? That would be some other time.
Yes. I'm working on that.
Okay, great. So if anybody has anything to show or would like to discuss further or have some suggestions, please. Okay, so maybe I will just use another 2 minutes of your time then to talk about something else. So we'll keep discussing this topic. I will post some replies, and please, if you have any ideas, follow up this discussion thread that Chad has started and share your thoughts. I would just like to say a few words about these webinars as we go forward. So we are expanding these, and you received my email about what the objective of these are now, which are a little bit expanded from what we had before, meaning that we will have presentations from different data generation centers, links data generation centers, that will tell us about data that they generated or maybe will generate it, that will go beyond gene expression data and definitely beyond L1000 data. And our goal is to get everybody interested in the analysis of these other data sets in the same way that there was that there is significant interest in this L1000 data. And maybe some of these other data sets will also offer opportunities to benchmark, say, L1000 against these other data types. So we will have, again, presentations scheduled that will be given by data generation centers, but in between we will also follow up with these kind of webinars in which we can discuss in details L1000 or any other data types that somebody is working with and would like to share their experiences. So thank you all for being here, and next week Laura Heiser from Oregon Data Generation Center will talk about some of their data.